Okay, in this second video on DNA replication, we're going to begin looking at some of the details of the molecular mechanism of replication. So in the previous video, part one, we looked at some kind of overview principles, how a genome is organized, how DNA gets packed into a nucleus, the semi-conservative model of DNA replication. And in the next couple videos, I want to start teasing apart all the little tiny details of proteins and what have you that get involved in actually replicating the DNA. You're going to want to watch these multiple times until they make sense to you. So let's start with a sequence of DNA called the origin of replication. So imagine this is a prokaryotic genome. They tend to be circular. The two circles on the left represent the two strands of DNA. So imagine that they are twisted together and base paired. There's going to be a single origin of replication on that chromosome, on that genome. In larger linear eukaryotic chromosomes, there are usually multiple origins of replication. But we're going to keep it simple and look at a prokaryotic genome. So this origin of replication, usually designated ORI, O-R-I, is a very short sequence, maybe half a dozen to a dozen base pairs, that is recognized and bound by a set of proteins called initiation proteins or replication initiation proteins. So when the cell, quote unquote, decides through signals that it's time to replicate its chromosome, it's going to express through gene expression processes, right, transcription, translation, these initiation proteins that will immediately seek out and bind to the origin of replication sequence. And by binding to that origin of replication sequence, they're going to disrupt the base pairing of those base pairs in that sequence. So what they do is, like you see on the right side here, is they're going to open up a little bubble where the DNA is not base paired anymore. And this is just, like I said, half a dozen to a dozen nucleotides in size. This is a very small stretch of DNA that is now single-stranded on either side. And that's important, isn't it? Because remember, we need single-stranded DNA. We need exposed, non-base paired nitrogenous bases to tell us what to do. That is going to act as our template. So this replication bubble is opened up first by these initiation proteins. So our initiation proteins, here we're zooming in on the bubble. What you see on the left and the right are supposed to be double-stranded regions, right? So the vertical lines up and down are representing the bonds, and those long, thin horizontal lines are the actual DNA, individual DNA molecules. So at the two X's, we have what are called replication forks inside the bubble. This is each end of the replication bubble where most of the action takes place. So the replication fork is where your double-stranded DNA becomes single-stranded DNA, where the two strands open up. Let's see if I can get a pointer here, where this is double-stranded DNA, and all of a sudden each strand is physically separated from the other because the initiation proteins here and here. This is where most of the action is going to take place in DNA replication. And we're going to see that the fork is going to move away from its current position. So this one here will move to the left, this one here will move to the right. And what I mean by move is there are proteins that are going to unzip or open up or pull apart, however you want to think about it, the double-stranded regions. They're going to disrupt the base pairing so that we have single-stranded regions of both strands exposed to act as templates for the new daughter strands to come in. So here's our replication bubble with our two replication forks. It was begun at the origin of replication by initiation proteins. So far, so good. If you need to pause it, play it back, whatever you got to do to get the terminology down, make sure you're tracking with me here. Next step is for a protein called DNA helicase to bind right at the fork. DNA helicase is all about untwisting the DNA. And by untwisting the DNA, it's going to force apart the nitrogenous bases, okay? Because the reason DNA is coiled into a double helix is so that those nitrogenous bases can actually reach each other through hydrogen bonding. And so by forcing apart or forcing the coil in the double helix to untwist, it's going to pull apart the nitrogenous bases and open it up at the replication forks. You can imagine this is going to take energy, and so this is very ATP intensive. It's going to consume a lot of ATP. And so each of these helicases will move in the direction of the double-stranded DNA, unzipping the DNA and opening up more and more, exposing more and more single-stranded DNA. That's the role of DNA helicase. So we've had initiation proteins to open things up initially, and DNA helicase is going to continue the process of unwinding and unzipping. Now, you can imagine if you've got these two strands that are twisted around each other. Try to picture maybe two strings that are twisted around each other. And you've got these long, long strings, a couple feet long, laying on the table in front of you, twisted around one another. Now, either in your imagination or actually go get some strings and do this, grab two of the strings with your fingers right in the middle, grab the two strings and start separating them, opening up your own little replication bubble. What's going to happen to the coils, to the twists in both directions as you're opening that up? They're going to supercoil. Those twists are going to get tighter and tighter. And unfortunately, the DNA would break if we didn't use something called DNA topoisomerase. Okay, so think of topography or shape, and isomer, changing shape, right? An isomer of something is a different orientation or structure of the same molecule, and ACE tells us it's an enzyme. So a topoisomerase is going to actually move ahead of, so let's just look at the right side fork. Topoisomerase is going to move ahead of the DNA helicase, so way down here someplace, I keep losing my pointer, way down here someplace, and it's going to nick the DNA, and it's going to allow the coils to unwind, and then it will religate the DNA. And it's going to keep jumping ahead of the bubble in both directions, untwisting that DNA that's becoming supercoiled ahead of the DNA helicase. So for example, in bacteria, there's an enzyme called DNA gyrase. It's one of these topoisomerases. We use DNA gyrase because these topoisomerases have to have two different activities, don't they? They have to have an endonuclease activity, meaning they have to be able to cut a DNA strand in the middle of the strand. That's what an endonuclease does. But then they also have to have a ligase activity to ligate it back together after the supercoiling has been relieved, after they've been allowed to unwind. So DNA gyrase has both endonuclease activity that cuts up the DNA, but then also ligase activity that puts it back together after supercoils have been relieved. Fluoroquinolone antibiotics, those are things like Cipro and Lexapro, not Lexapro, Levo, right? Lexapro is an anti-anxiety drug. But things like Cipro, you're all familiar with Cipro, right? Ciprofloxacin. Fluoroquinolones, like those drugs, go after DNA gyrase specifically, and they knock out the ligase activity, but they leave intact the endonuclease activity. So think about what happens then. The bacteria attempt to replicate their DNA so that they can grow and divide, and they end up chopping up their own chromosome into pieces, and they can't put it all back together again. Very, very clever approach for us to be able to inhibit them, and it's a drug or a class of drugs that specifically goes after the bacterial DNA gyrase and doesn't touch our topoisomerases, so it's highly specific. So make sure that makes sense to you, this whole idea of supercoiling because of the helicase and the role of the topoisomerase is to relieve that supercoiling. Now, another problem we have is that we've got this single-stranded DNA that's opening up, and realistically, all these base pairs that are stretched out apart from each other would really like to be back together. That would be, they're sort of attracted almost, you could think of it almost magnetically. The hydrogen bonds are drawing them back together. And so temporarily, at least, until we can get our DNA polymerase in here to make another strand, we need to protect the two single-stranded regions so they stay open and available. And we use something called single-strand binding proteins, or SSBPs. So these single-strand binding proteins stabilize the single-stranded DNA in this open conformation long enough for the DNA polymerase to come in and begin reading those single-stranded regions and creating the new DNA. So I think that's as far as I want to go in this video. Go through it a couple times, make sure it makes sense, pull your book out and go through it up to this point. Don't jump into the next step with DNA polymerase until all of these initial steps of initiation, helicase, and single-strand binding proteins, topoisomerase, all these steps make sense. Make sure you can visualize what all these different little images in the diagrams are representing, that it's not just a yellow blob, but it's an enzyme that's grabbed onto the double-stranded DNA at the fork, opening it up, unwinding it, etc. Think your way through all this before you move on to the next video, and I think you'll find the next video will make a whole lot more sense if you're willing to do that. And as always, let me know what questions come up as you're working through all this.